Hello, hello, hello. How are you, everybody? Here we are again on a new topic today, which you should know, of course, constitutive versus inducible enzymes. A few points in the concept of these two categories of enzymes, of course. Let's go. What are these constitutive enzymes? These enzymes are produced at a constant rate and are always present in the cell. Their genes are active always when they are always active in the body, regardless of the metabolic state or environmental conditions. And they are essential for basic cellular functions. These enzymes are present there to catalyze reactions what are required every time for basic cellular functions like energy production, which is incessant as far as the cell is living. They are they are often involved in fundamental processes such as glycolysis, the citric acid cycle, the link reaction, all these pathways or chemical reactions that are involved in what we call cellular respiration. Examples including enzymes like hexokinase and pyruvate kinase, which are crucial for energy metabolism. The other category, the opposite, inducible enzymes. Contrast to inducible enzymes, these are synthesized only when needed. The genes are active only when these enzymes are required in the body. Typically, in response to specific signals, the enzymes are produced whenever there is signal from the environment or environmental signals. This allows the organism to adapt to changing conditions by producing enzymes that are necessary for utilizing available resources. When resources are available for catalysis, these resources or substrates attach on the specific region in the enzyme and activate that enzyme so that transcription translation happens and the enzyme is produced. A classic example is the beta galactosidase, this enzyme in Escherichia coli. Beta galactosidase. Uh, in this enzyme, it has beta galactosidase gene in Escherichia coli, which is produced only when lactose is present in the environment. Sorry. Therefore, lactose is a signal from the environment. If this bacteria is put in a medium rich in lactose, lactose serves as signal for uh, serving as transcription factor for the gene that produces beta galactosidase so that beta galactosidase can be produced, which is involved in metabolism of lactose. Mechanism of regulation. How do we regulate these two groups of enzymes? Constitutive expression. One is mechanism of constitutive expression. The genes encoding constitutive enzymes are continuously active, driven by constitutive promoters that initiate transcription without external signal. They don't require external signal for the gene to be expressed. Therefore, there are constitutive promoters which always go and attach on that specific gene that produces the constitutive enzymes so that the enzymes are produced. This ensures a steady supply of essential proteins required for survival and basic cellular function. You see, then stop, continued steady supply of essential proteins possible because the genes that encode for constitutive expression are the genes that are promoted or made be expressed by constitutive promoters, not external environmental signals. Inducible expression. The expression of inducible enzymes is tightly regulated. These enzymes are typically off by default and are activated by specific inducers, which can be substrate or other signal molecules in the environment. For instance, when lactose is present, it binds to a repressor protein, allowing the transcription of genes needed to metabolize the lactose. You see, repressor protein to which lactose attaches so that the gene is the repressor protein is of the gene. Their, the gene that produces the enzyme that's needed for lactose metabolism. This is the expression of inducible genes. And thank you very much. Finally, before I say goodbye, of biological significance of these two constitutive enzymes, they play a crucial role in maintaining cellular homeostasis and are vital for processes that are always required for cell survival, such as energy production, as we have metabolic pathways. Uh, we already mentioned involved in many metabolic pathways that are essential, always active, and a steady supply of products in the body. Inducible enzymes, these enzymes provide flexibility and adaptability to the organism, allowing it to respond to environmental changes and utilize available nutrients efficiently. This adaptability is particularly important in fluctuating environments where resources may vary, of course. Therefore, this is the significance of these two groups of enzymes. And thank you very much. Bye bye. And keep on please subscribing, sharing, and liking my videos. Thank you. Ciao.